Hi everyone, I am going to explain about the topic steps involved in drug development. Before going to the topic, what is drug development? Drug development is the process of bringing a new drug molecule into the clinical practice. It involves a series of steps that brings after a potential drug candidate has been identified through the process of drug discovery. These stages take drug from preclinical research to clinical trials and ultimately to the regulatory approval. And drug discovery process includes first stage known as discovery and screening, and the second step lead optimization, which is the drug is converted into a lead, and uh, it undergoes pharmacodynamics, pharmacokinetics and clinical trials after NDA approval. So first process in drug development is the preclinical research. It involves three steps: target identification and validation, hit discovery, hit to lead optimization. Target identification is the researchers identify a biological target. It may be a protein or gene that is implicit in a disease. Hit discovery. Chemical compounds are screened to find molecules that interact with the target. This process takes a high throughput screening method for the discovery of the hit. Then the discovered hit is converted into lead, and the compounds are refined and optimized for the potency, selectivity, and other pharmacological properties. So preclinical research is nothing but it is the phase of drug development, takes the drug in human clinical trial. Before it goings to human clinical trial, it involves a laboratory practice, and the drug is tested in the animals to assess the safety, efficacy and pharmacodynamics and kinetics and toxicology of the drug compound. The primary goal of the preclinical research is to gather enough data to support the initiation of the clinical trials in humans. And the second step is safety assessment. Researchers investigate the potential toxic effects of the candidate compound on various cell types and tissues. They start doing research. Their aim is to identify any harmful effect that could arise from the drug's interaction with biological systems. And the third step is pharmacodynamics and pharmacokinetics. Pharmacodynamics is nothing but how the drug reacts with the body, including its mechanism of action and cellular response, etc. Pharmacokinetics is the body how reacts to the drug, and the drug they focus on how the drug absorbed, distributed, metabolized and excreted by the body. This information helps to determine the appropriate dosage and dosing frequency. And the fourth step is efficacy studies. Researchers assess the drug's effectiveness in its laboratory settings using cell culture or animal models that mimic the disease being targeted. These studies help to determine whether the drug has the desired therapeutic effect on the disease. And the fifth step is formulation development. The drug is formulated into various dosage forms, such as it may be a tablet, capsule, or injection, to ensure the stability, appropriate release, and effective delivery in the body. And the sixth step is animal studies. The drug is tested in animal models that because of the animals resemble close to the humans. So researchers evaluate the drug's efficacy and safety in these models and observe its effects on disease progression and potential side effects. After that, they cure that and will test in the humans. Seventh step is the toxicology studies. Researchers assess the potential toxic effects of the drug candidate on different organs and systems. These studies helps them to determine the safe dosage and provide data on the potential effect. So eighth one is a good laboratory practice, which is very important. Preclinical studies are conducted according to their good laboratory guidelines, which ensures the accurate, reliable and ethical experimental practices. Data generated in compliance with GLP standards are crucial for the regulatory submissions. Ninth step is data compilation and analysis. All the data collected during the preclinical research are analyzed and compiled into the comprehensive report that supports the safety and efficacy of the drug candidate. And tenth is the regulatory submission. This results preclinical studies are included in the regulatory submissions, such as it undergoes the investigational new drug application, which is submitted to the regulatory agencies to gain the approval for conducting human clinical trials. The success of preclinical research is a drug candidate will proceed to the clinical trial. The data generated during the phase provide critical insight into the compound's potential therapeutic benefits and potential risk, guiding the decision making process for the further development. So overall, it undergoes three process, which is in vitro and in vivo testing, which is done in the laboratory condition. It involves basic research and early discovery and preclinical trial. After that, it undergoes in investigational new drug application. When they approve, it moves to the further step, human testing, in which is the clinical development or clinical trial. It undergoes three phases: phase 1, phase 2, and phase 3. In phase 1, small amount of drug are tested on the healthy persons, and in phase 2, a large amount of drugs are tested on the patients, and in the phase 3, large amount of drugs are tested on the both healthy persons and patients to note their safety, efficacy, and effectiveness, toxicology, etc. After that, the new drug administration or application will approve the drug, which involves FDA review or data review. So FDA approve, once the FDA gives approval to the drug, then the drug will go to the market. After that, the drug go to the market, it undergoes the surveillance of the post marketing market monitoring. So once the drug is released, the government will monitor how the drug is delivered. So that's all about drug development process. Thank you.